started. Thank you all for joining us for this installation of the Virginia Tech Life Sciences Seminar Series. I'm absolutely thrilled to introduce today's speaker, Professor Rommie Amaro from the University of California, San Diego. Dr. Amaro is a distinguished professor in theoretical and computational chemistry out at UCSD. She began her educational training by completing a bachelor's degree in chemical engineering, followed by a PhD in chemistry. And then she was a postdoctoral fellow with Andy McCammon at UCSD. Andy, of course, is famous for being one of the pioneers in protein simulation. And I'm pleased to say Dr. Amaro has continued on in that vein of being a groundbreaking scientist in her own right. And so she started her independent lab then after completing that fellowship with Andy in 2009 at UC Irvine, and then moved back to UCSD where she has been ever since. She has won numerous awards, more than I could even take the entire hour to probably tell you about, but an NIH New Innovator Award, a PCASE Award, several from the American Chemical Society, and most recently, probably the headliner here in the last couple of years, the Gordon Bell Prize for her work on COVID-19, which we affectionately call the Nobel Prize of Supercomputing. So it's a really huge deal and certainly well earned, and you'll hear the details about that today, so I won't steal her thunder on what that project was about. She's done a large amount of service to the scientific community. She's been an editorial advisory board member to all of the prominent journals in computational chemistry, and she's currently an associate editor at the Journal of Chemical Information and Modeling. She has 161 peer-reviewed journal articles, 6 books and book chapters, and she's been an impressive CV. I was reviewing it, it's very impressive.
Wow, you really did your homework, Justin. My goodness, okay.
And steadily funded by all, NIH, NSF, and many others. She has done incredible work in complex biomolecular systems. Her papers are the ones that always I take notice of because, for those who read her work, please do, because they always ask really important and specific scientific questions and really use the best methodology. And so with that, I'm going to turn the floor over to Dr. Amaro. If you would please join me with a digital welcome out toward, point yourselves in the direction of San Diego, wave hello, and give her some digital applause. And Dr. Amaro, the floor is yours.
Oh my goodness, Justin. That was probably the best introduction I've ever had. Thank you, really, that was terrific. I'm so happy to be here visiting you virtually to tell you all about our work using computational microscopy as a tool to study SARS-CoV-2. We'll get started. Okay, so in my group, so I'm a computational and theoretical biophysical chemist, and so everything we do in my group is computational and theoretical, so I don't have a wet lab. But nowadays, I like to think of the main method that we use is something called molecular dynamic simulations, and I like to think of it as a computational microscope because that really is exactly how we try to use it. So nowadays, this microscope is actually quite powerful, and it allows us to bring together multiple different types of experimental data sets together to build these highly detailed 3-dimensional models of biological and chemical systems. So for example, with a virus, what we've been able to do that I'm going to tell you about today is we're combining different types of structural biological data techniques, so cryo-electron microscopy, X-ray crystallography, and tomography. And we combine those structural techniques together with glycomics, for example, to talk about glycosylation and so forth, lipidomics, as well as genomics. And so we build these highly detailed sort of large models, and then we approximate those models down to their many atoms. And once we have that system built, we have this potential function that I'm showing here. And this basically describes all of the interactions that the atoms have with other atoms in the system. And then all we're doing is basically integrating Newton's equation of motion over time, so we can understand the sort of the atomic level dynamics of the system as it evolves in time. And we perform this numerical integration millions and billions and trillions of time steps to build up a trajectory or a dynamical understanding of the molecular piece parts, if you will, of the system. And nowadays we use, the computers where we run where these microscopes run, sort of under the hood of these simulations are these really powerful supercomputers. So for the work I'll tell you about today, we primarily use TAC Frontera, which is a machine in Texas, as well as the Summit supercomputer at Oak Ridge National Lab. These are two of the top 10 fastest machines in the world. And, owing to their power, we are able to not only build these data-centric systems, but also simulate them in order to extend our understanding of these systems and experimental data. Okay, so it was about mid-February of last year when things really changed for the world and also for our group. So we had been working, until then, I had been working for many years to study the influenza virus. And in around mid-February, this turned out to be a pretty important week for us. This was the week that we published, after a 6 or 7 year long labor of love project, an all-atom molecular dynamics simulation and analysis of the influenza viral envelope, which is shown here on the cover of ACS Central Science. And we were so pleased that that finally got out, because it was groundbreaking in the sense that it was one of the largest biophysical simulations that had been completed, and it allowed us to understand things about the flu and how it bound to receptors on the host cell. But that same week, things were changing in the world. So what I'm showing here are the total confirmed COVID-19 cases for the early months of the pandemic from January through May of last year on log scale. And you can see in the United States in mid-February, there were really still very few cases, maybe about a dozen circulating cases here. But we knew that the virus had already escaped mainland China. And we knew that, for example, in the Lombardy region of Italy, the virus was just it was looking very grim. So that was when we started to think, well, maybe we need to sort of shift gears here from what we had been studying with the flu and think about studying this new virus. And so that same week, something else really important happened, and that was that the group of Jason McClelland, together with collaborators from the NIH, put into the bioarchive the first cryo-electron microscopy structure of the SARS-CoV-2 spike protein. And this was a real game changer for us, because for our types of methods, we really ideally rely on structure. So this gave us sort of the major sort of starting point that we needed to try to work with this virus. So this was a day that really a lot of my group really pivoted towards SARS-CoV-2. And these three folks here, Lorenzo Castellino, Ziad Gayeb, and Abigail Dahmer, really led that charge. They're still leading it, but they really led the charge in the early days. And so just a little bit of background on SARS-CoV-2. So I'm sure everyone here is already, unfortunately, well aware of it. This is an image of the virus created in my group. But basically the virus, it sort of looks like a golf ball with spikes sticking out of it. And these spikes are, in fact, we call them the spike proteins. And we care about these spike proteins because they sit on the outside of the virus. And so they're the first point of contact that the virus has with the human cell. So they play a really important role in the initial infection event. These spike proteins are also highly antigenic. That means that our immune system, responds to them in mounting the immune response. And so, not surprisingly, these are the molecules that are inside all of the approved vaccines, and hopefully most of you have already been injected with those. So this talk today is really going to focus on the initial infection event and trying to understand better what is happening and sort of the detailed mechanisms of how the virus actually latches onto the host cell. And so in order for infection to happen, the spike protein, which I'm showing here in white, needs to interact with or make contact with something on the host cell that allows it entry. And for SARS-CoV-2, that bit is this yellow molecule I'm showing here. Here's a zoom in of these interactions. This is the yellow, the human receptor that the virus interacts with is called ACE2, or angiotensin converting enzyme 2. And so, this blue bit in the middle here, I'll draw your attention to. This is called the receptor binding domain, or the RBD. And I'm going to keep coming back to that throughout the talk, because this part of the spike protein is really the key surface that needs to make that contact, or that handshake, if you will, between the virus and the host cell. And that's what really sort of, precipitates infection. So in the talk today, I'm going to discuss our efforts to understand, again, sort of like the dynamics of the spike protein, as well as its interactions with ACE2, and sort of then I'll touch on at the end our larger scale efforts to try to understand the molecular piece parts inside of like these much more complicated sort of whole virion environments. Okay, so as I mentioned, so that the first piece of data that came out that was really important for us was this cryo-EM structure of the spike head by Jason McClelland, which was published in Science in mid-March of last year. And this paper amazingly has been cited, I think, like over 5,000 times already, which is just, you know, enormous the impact that it's had. So this is the structure that the group presented. You can see here very clearly the head of the spike protein. So these spike proteins are pretty complicated molecular machines. They are trimers, so they're made up of 3 identical protomers, or sort of subunits. And I'm showing one end-to-end subunit there. The cryo-electron microscopists, what they can resolve is what we call the head of the spike. Okay, and this is what I'm showing here. I blurred out what they can't see, which is the stalk and the cytoplasmic tail. I'll tell you about that in a little bit. But so they published this first structure that actually had the receptor binding domain that I talked to you about in the last slide in an up or open conformation. So this little green bit here. A few weeks later, another terrific structural biology group, the group of David Wiesler at the University of Washington, published a second study that largely recapitulated the findings of the open state that Jason and his group had found, but they also used averaging, symmetry averaging, to create a model of the closed spike as shown here. And so we took both the open spike and the closed spike that they determined from cryo-EM, and then with computing, we completed them. And so one of the challenges, and I'll touch on this again in a bit, is that you know, as powerful as these experimental techniques are, for example, cryo-electron microscopy, there are still things that they can't see. And where they're limited tend to be if there are parts of the protein or the system that move a lot, that are highly dynamic. Just like taking a Polaroid picture, in some sense, a picture of something, these bits that move around too much can be sort of blurry or not in high resolution. So we can use computing to model in those missing pieces, and that's what we did in order to create sort of a complete model of the spike head. But what we really wanted to do was to model the entire spike. So we took the spike head, and then we also modeled the stalk. So the spike head is basically attached to the viral membrane by a trimeric coil-coil bundle. It's basically a tether. And this is the transmembrane component of the spike. And then it also has this little bit sticking in on the inside that they call the cytoplasmic tail. So we were able to model the full sort of end-to-end structure. And this is what it looks like, you know, in full. So the other thing that experimental structural biologists can't see are these things called N and O linked glycans. And so N and O linked glycans are, they're an important post-translational modification of proteins. And they do all sorts of things like help proteins fold and help with sort of shuttling of proteins, and they're involved in, you know, various biological phenomena. But they're very flexible, dynamic entities. And so the cryo-electron microscopists and structural biologists, they can't see them. They can't ever really resolve them very well. So this is a place where computing and modeling has a lot to give, because what we can do is take, for example, the glycomics data, and there was this beautiful study published by Max Crispin and Jason early on in the pandemic. I think this came out in April of 2020, that gave researchers using mass spectrometry and glycomics approaches, this basically gave researchers the detailed molecular recipes of each of these, the molecular composition or the recipes of each of these N-linked glycans on the spike protein. So I mentioned that the spike is a trimer. Each protomer of the spike has 22 of these N-linked glycans of varying composition, as you can see here, and at least 2 O-linked glycans. And so our models actually can, you know, we rebuild these into our models in order to understand what it looks like. And so we build these models, and then what we're doing is basically simulating the motion of the system over time. And so here you can see what you see here, this sort of the movement, this sort of wiggling and jiggling of these atoms, that's the output of the molecular dynamics simulations. And so for these simulations, we had, and I'm just going to loop through this movie again, we created different systems. We had the RBD in the up conformation and the RBD in the down conformation, or the open and the closed conformations, as I was talking about. These systems have about 1.7 million atoms each, and we use the Charm 36 force field with NAMD2 on that TAC Frontera supercomputer in order to simulate several microseconds of dynamics for each of these systems. And one thing that I want to just pause and say here is that, when you're looking at this, it may be tempting to say, oh, it's a pretty movie and it's a pretty picture, but it's important to remember that these simulations are numerical statistical mechanics. And so that means that these simulations, these calculations are performed, in accordance with sort of the laws of thermodynamics and so forth. So what we can do is take out of these simulations, we can basically take averaged microscopic properties and directly connect them to, or to compute, experimentally testable macroscopic observables. So we can connect, because these simulations are theoretically rigorous, we can connect the output directly to things like binding free energies, heat capacities, and so forth. And so the real power of these methods, in my view, is not only the ability to sort of synthesize these cohesive data sets into a pretty picture, but then, even more powerfully is to actually begin to understand their functional dynamics and to have that close interplay with experiment so that we can, develop new hypotheses and understand how these systems are working. So what did we learn? So the first thing that, was really evident, and I think one of the reasons why, this work was so well received, was that we showed the world for the first time what the full length, fully glycosylated protein actually looked like. And so on the left, I'm showing the full length protein. This is the spike protein with its tether and the viral membrane. And this is essentially what the structural biologists see. They see the protein, and that is so important, right? Because the protein does a huge amount of work and has obviously core functions. But again, if you look on the right, this is what that same model looks like if we actually could see the glycans. And so these blue fuzzy bits are basically the glycans. And so each of these little balls of fur or fuzz represents a composite image of many snapshots taken from, so it's one glycan taken with snapshots over 1 microsecond of dynamics. So what you can see, I think so clearly or so effectively, is how each of these glycans sort of sweeps out a volume of space. And these are fast dynamics happening on the nanosecond time scale. And when it does that, so they're sort of moving around and they create this sort of dynamical shield. Literally, they call it a glycan shield. And that shield hides the virus and the foreign matter, such as the spike protein of the virus, from the human immune system. And as you can see, the SARS-CoV-2 spike protein is fairly well covered. So it's very good at hiding itself from the immune system. And this type of information of understanding where the glycans are and what they look like, this is really important for vaccine and drug design, because in order for our immune response to be mounted, typically parts of the protein need to be recognized, or these epitopes need to be recognized or unveiled. So our simulations also showed why the spike actually opens and closes. So on the left, I'm showing a picture of the closed spike from the top-down view. And you can see in blue, again, the glycan shield, and in dark blue. And in this light blue color, that's that receptor binding domain that I've talked about. I'm trying to keep the coloring the same. You can see how in the closed state, the receptor binding domain is sort of covered up by these sugars. And from the side view, so this is the side view of the spike in that closed conformation, you can see how it's barely visible. It's sort of tucked down underneath. So this is what we call the defending mode of the spike, or it's hiding. It's doing very good at disguising itself. In contrast, on the right, I'm showing what the open spike looks like. And you can see very clearly. So in the open spike, this blue receptor binding domain is lifted up out of that glycan shield. And here's the top view and the bottom view. You can see how it's literally just popped up like a snorkel. And so this is really important because this piece right here that's exposed in this open state is the part that needs to bind to ACE2 to infect the cells. So this is really sort of the attack mode of the, or the infection competent mode of the spike protein. And so if you don't have the glycans there, if you can't see them, which experimentalists can't, you don't have the sense for why it's undergoing this motion. And it's very clear once you consider where the glycans are, that it has to lift up that RBD in order to actually infect the cell to get above that glycan shield. Glycans also help, they can actually help escape the immune response. And so this is something that we know from years of influenza and other viral research, that over time, and we've heard about these different mutations of these variants of concern. The virus has the ability to mutate its its core sequence, its amino acid sequence, primary sequence. And in some instances, when it does that, it introduces a new glycan. Okay, so these glycans are encoded in these things called sequons. It's a small stretch of amino acids. And when it creates one of those, it signals to the glycosylation machinery, add a glycan here. And so, we work together with Rino Rapulli, again, and Jason McClellan, where they had passaged viruses, the SARS-CoV-2 virus in potent polyclonal sera. And over some months, what you'll see when that happens is the virus continues to evolve, and it actually evolves into an immune, it can escape the immune response. And what we saw in this instance, which we thought very interesting, was that this virus over time and under immunogenic pressure created a large insertion in the N-terminal domain. I'll just note, we haven't seen this in circulating viruses. These are more extreme experiments that are sort of lab performed situations, but it shows what the virus is capable of. In this case, it's capable of bringing in a very large insertion that actually does involve a new glycan. And so, here is a side view of this antibody. I'm showing in gray here. This is an antibody, a neutralizing antibody, which should bind and neutralize this part of the spike protein. But you can see here when we model in this new glycan, it basically, if you insert this branched chain, molecule into this region, it completely obliterates the ability of these neutralizing antibodies to bind. And that's what we found for this particular strain, that the addition of the glycan could actually evade the immune response. Okay, but then beyond shielding, something interesting really popped out in our study, and that was that when we looked closely at the system, when we started working with it, and I'll note this is a fantastic collaboration with Elisa Fada at Maynooth University. When we looked at this closed state from looking top down on the spike, one thing we noticed was that there was this one particular glycan that was always sticking sort of outward into solution. Okay, then we looked at the open structure. And what we saw was that, okay, two of these glycans were sticking outwards, just like we saw, but one of them actually was pointed inwards. And so we took a closer look. And so now what I'm showing you is another view of the spike protein head. In blue, again, is this receptor binding domain that I keep talking about. In red and gray are the other domains of the spike head. And then here in navy blue and green are these glycans. And these are two glycans, N165 and N234. The dark blue bits are the part of the glycan that the cryo-electron microscopists could actually resolve. So in this instance, they could actually resolve the first sugar unit. The green bits are the parts of the sugars that we had to actually model. And what we found was that when we rebuilt those sugars, into their full complexity, that these sugars appeared to fill in the void space that was created when that RBD lifted up into that open conformation. So it occurred to us that these glycans were sort of providing like structural stability or support to the spike protein, that they were sort of acting almost like a kickstand on a bicycle. And so then we thought, okay, well, what happens if we would mutate these two residues. If we introduce an alanine at these positions, it will abrogate the, there won't be glycans added at that site. And our hypothesis was that then if we ran the dynamics, or just in general, this receptor binding domain no longer supported would collapse down, and the interaction with ACE2 would be reduced. So we created the Insilico mutations and we ran them, and we did in fact see this receptor binding domain collapse down. And then I called up, somebody thought, okay, maybe there's something to this hypothesis. So then I called up Jason McClellan. I had never met him before. I figured he was very busy because he's just like this amazing spike, protein, and he's a co-inventor of the Moderna vaccine, this 2P mutation guy. And but he was just terrific collaborator. He said, absolutely, yeah, I'd love to test these mutations. Let's do it. So he and his group members created a biolayer interferometry assay that allowed them to basically see what would happen with the interaction with ACE2 when they introduced these mutations. And so with 165A and 234A, this is our control, we see that when we have these mutations, the interaction with ACE2 is reduced. So this supported our hypothesis and allowed us to sort of understand or learn that these glycans, beyond just the shielding that they ordinarily do, that there were two glycans in particular that were actually participating in the infection mechanism itself of the SARS-CoV-2 virus. And this was really cool. It sort of gave us a new understanding, not only of this virus, but also of the ability of glycans to participate in infection mechanisms for these types of viral machines. Then, beyond shielding, we wanted to, in that study, what I was talking about was we had basically simulated sort of these different endpoints. So we had the closed structure from the cryo-EM and the open structure from the cryo-EM, and we were just looking at sort of the low level, sort of short time scale dynamics around both of those two states, which had been, resolved with cryo-electron microscopy. But, we can also use simulation, or we would like to, just to understand what does that actual opening transition look like. And so, unfortunately, it's very difficult to do that using conventional molecular dynamics. It would take some 800 years to do, because we know experimentally now that through FRET experiments from the Moffett's lab, that actually that opening motion happens on the seconds time scale. So in order to actually get these detailed views at the atomic level, it would take a very long time to simulate. So here we started a new collaboration with Lillian Chong at the University of Pittsburgh. And she's a fantastic method developer and computational chemist in her own right. She's been working for some years to develop, together with Dan Zuckerman and others, this something called the weighted ensemble MD. And so this is sort of what we call an enhanced sampling method. And it's basically a way of sort of speeding up molecular dynamics, but it does it in a way that it stays true to its sort of statistical mechanics. So we can go back and still make these rigorous connections with experiments. And so it's very powerful. And so, because what happens often in molecular dynamics simulations is that if we have this free energy of the system, and we spend a lot of time, in these structurally characterized low-lying states, right? But most of the functional behavior, is outside of these states, and they have to go over barriers. So her method allows us to basically enrich our simulations towards these states where interesting things happen, such as the opening of the spike head. So we partnered with her and her team to use weighted ensemble simulations of the spike head in order to sample this transition. And so with her method and using another partition of this supercomputer called Longhorn, we took over 100 of these NVIDIA V100 GPUs, and we're able to run them for about 3 or 4 weeks straight, which was awesome. And using her method, we generated over 300 of these opening pathways. And again, why that's important, I'll just say, if we wanted to use standard conventional molecular dynamics like I talked to you about in the first part of the talk, it would take hundreds of years to actually, on a single processor, to actually get to that same state, because the biological timescales are so slow. So, so what was really cool about this was it basically allowed us to monitor sort of the opening of the spike receptor binding domain. And so here I'm showing in the spike core, and I'm showing sort of the range of transitions that sort of the central part of that receptor binding domain undergoes. So in the closed state, the central sheet here is represented by this hot pink ribbon. And this aligns with the 6VXX closed structure I was telling you about from Wiesler. And the open structure is actually this one in light blue, which is 6VSB. Since then, other cryo-electron microscopists have discovered even more open than the original open. So we call them sort of wide open structures, such as 7A95. And we can use simulations to basically interpolate in a rigorous way sort of through this transition. So this is sort of what that opening process looks like. You can see how it will, again, just allow us to, in an unbiased manner, sample this opening pathway from the closed state, and there it goes to the open state. And this was work led by Tara Stain and Shirley Ahn, which was recently published in Nature Chemistry. But what was so cool about this was that, so I'm showing the glycans again in blue. But there's one glycan in particular that I want to bring your attention to. This is this hot pink one here in the middle. This is N343. It turns out this N343 basically looks like it's acting like a molecular crowbar, and at some point helps to lift and prop up that RBD into the open conformation. Here is another view of that. Hopefully this one plays. Sometimes this movie gets fussy. It got fussy. So, so basically, what was so interesting about this was, let me see, do I have it on the next slide? Not really, but so what was so interesting about this, I mean, I'll go back to this one. Sorry for my movie failure. When we had done our initial study and we looked at the closed state and the open state, when you look at this particular glycan, it looks the same. It's just like in the closed state, it's sitting there, it's helping to shield. In the open state, it's sitting in about the same spot, and it's just looking like it's helping to shield. But when you look at the functional transition, when you actually look at the mechanism of opening, you see what an active participant this glycan actually is in the opening mechanism. And so that to me was so cool, because it shows the power of these types of enhanced sampling simulations to like reveal something that you really otherwise wouldn't see. You know, even when it's sort of these images are right before your eyes, you don't know it because you haven't seen the dynamics. And so another thing that was interesting was, while this paper was in the bioarchive, we serendipitously connected with Joachim Frank and Evan Seitz at Columbia University, and they had been using a method called Manifold EM, which is an AI-based method. So, you know, Joachim Frank got the Nobel Prize for cryo-electron microscopy method development in 2017 or so, some years ago. And he's been developing methods to try to go beyond looking at sort of just the single high resolution structure that everybody focuses on, and they get their, you know, the structural biologists, they get their science paper and then they kind of move on. There's, you know, they have these, you know, they have hundreds of thousands of images, many of which, you know, to date, they pretty much throw away. So there's like a lot of people trying to figure out what to do with that data. He's one of them, he and his team, and they have developed this method that tries to allow them to sort of mine what types of other structures are in these data sets. And so I'm going to play that movie again. So on the left is our weighted ensemble trajectory. You can see it opening, and the right, and in the middle and the right, these are the manifold EM sort of images that they get when you look at the other data images in the cryo-EM data set. So what this tells us is that our simulated opening pathway, and we did this sort of more rigorously in the paper, but it aligns very well with what they see in these other structures that are present in the set. So it helps us as sort of indirectly validate the actual pathway of opening that we developed with the weighted ensemble approach. And so then Jason McClellan and his team actually were kind enough to do even more experiments for us. They created a new series of mutations that included not only the glycan I told you about, but other residues also on the receptor binding domain and nearby that we thought were important. And what was so interesting was that we found that this N343 343A, the glycan which we ignored in the first study because it didn't look like he was doing anything, actually shows the biggest reduction of ACE2 binding, much bigger than 165 and 234. So that was really cool that not only these two glycans are stabilizing it up, but that this 343 actually acts like a gate. It sort of initializes that down to up transition of the receptor binding domain. And since then, other groups have shown that it's actually, this glycan is really essential for cell infection. So it's really cool. Okay, so then moving beyond the spike, what we really want to do is get a better view of like how this thing is actually infecting host cells. So to do that, we also want to study the flexibility of the ACE2 receptor. And so we built models of ACE2, similarly with similar complexity of the spike protein, having all the glycosylations and a plasma membrane. This was led by Amelia Barros in my group, who's now moved on to a postdoc. ACE2 is also glycosylated, less so, but you can see those glycans here where they're added and so forth. And we ran dynamics again, microsecond scale dynamics. And what we found here that was interesting actually was that this molecule, this ACE2, actually exhibits a remarkable degree of flexibility relative to the membrane normal. It basically can just completely bend over and almost, in fact, some of these glycans actually touch the membrane and interact with the membrane. And it sort of, even on the nanosecond time scale, can sort of wobble back and forth, which was shown here. And so we think that this is really helping in the mechanics of spike infection. So then what we did was we combined our full length spike that had the RBD and the up conformation with ACE2 in order to create a parallel, sort of two membrane system, right? So this would be the equivalent of, the virus sort of coming in with a spike protein and actually making that infection event with a receptor on the host cell here. And these membranes also have different compositions because this would be the plasma, the cell membrane for humans, and this would be more like the Golgi or the viral membrane. And so these simulations have about 10 million atoms each, so going up about 5-fold in size. And they're a little bit more complicated because of boundary conditions and so forth. I just leave some of the more technical details here. What we actually had to do was insert an OMP-G pore, in fact, into these membranes in order to work out the water dynamics so that water could freely pass and we don't get any sort of weird pressure effects inside our cell. But it doesn't change the dynamics of the protein itself. and it allowed us to sort of simulate the system. That was actually Saima Khalid at Oxford University now helped us with that addition. Oh, and so this work has largely been there's a lot of science here that we're still actually taking out, and we're looking at correlated network analysis and so forth across this interface. But a first description of this work actually was presented in this in our Gordon Bell paper, which is shown here. And so what we can do is actually, as I mentioned, we're sort of simulating these dynamics, and we can get these views then not only into sort of the interactions right at the interface, which is important also and interesting. It's possible to develop therapeutics that could disrupt that, but also it gives us new views into stalk bending. And Gerhard Humer and others have shown, Martin Beck, the tomographers, showed that actually there's a remarkable number of movable joints in these systems, particularly in the spike protein, not only in the receptor binding domain and the S1 and S2 subunits, but also in this stalk, which is very difficult, not possible yet to resolve in high resolution, but they can see with tomography some bending. And but one of the issues that we have methodologically is that as we get bigger systems, it's harder for us to sample because there's more atoms and more degrees of freedom. So here we partnered with Arvind Ramanathan and Shantanu Jha and their teams. They have developed something called Deep Drive MD, which is shown here. There's references if more interest. They basically developed an AI-based, it's an adversarial autoencoder that basically took our weighted ensemble trajectories. Oh, and by the way, this was the movie that didn't play earlier. Oh, except that it doesn't have the pink glycan. It doesn't show you the pink 343. Anyway, so it's sort of similar. 343 is right in there. Anyway, so they basically took our weighted ensemble simulations and had their method sort of learn what the interesting phase space was for opening. And then we used that analysis to reseed new replicates that were then sent off to get more sampling. So we do sort of like iterative sampling of these MD trajectories, where our restart configurations are sort of help guided by these machine learning based sort of informed methods. And sorry, that gets a bit technical. So for those outside the field, but like for those in the field, it might be something of interest. And this is something we keep working on. And why we like this is because it helped us essentially sample more efficiently these interesting regions of phase space, which is a challenge when you do get to these bigger systems. And so what does that allow us to see? So here I'm showing two different sort of representations of the spike in conjunction with ACE2s, right? So on the left is just like a drawn schematic. Here's ACE2 as we've modeled it. Here's the spike. Here's the spike head. This is the RBD and the up conformation. And this is the stalk, and this is the transmembrane domain. So Gerhard Humer and Martin Beck, they basically defined, they were nice to define some nomenclature for how we could talk about these different parts of the spike. So they call this the hip, the knee, and the ankle for this bendable bits here, because it kind of does look like a leg. And so so we can actually see, in our simulations, we see a great degree of bending at the ankle. We also see a significant degree of bending at the knee. You can see that here, where it's maintaining contact the whole time, perfect contact to ACE2, which is interesting. And then we also have significant bending at the hip. And then finally, the spike head itself also undergoes tilting and sort of also relative to the whole stalk. And you can see that here. So this allows us, again, we can really start to dissect these sort of detailed dynamics of the system as it's undergoing, sort of a real, hopefully the first steps onto self-fusion. And, we continue to work on that. And then the other thing, as I sort of mentioned right at the beginning, I mean, I'm very interested in sort of pushing molecular simulation to the mesoscale, where we can have these really highly detailed models of very complete biologically realistic systems. And so, so we were really excited to see the beautiful set of tomography data coming out of multiple labs late last summer in August. They were able to visualize using cryo-electron microscopy and Fib milling real sort of like the viruses in cells. And so this gave us really beautiful views of the size of the viruses, as well as the patterning of the different spike proteins. And here's where they also saw that there were bending at these joints, right? Which I just showed you in these molecular simulations, but here you can see static snapshots at low resolution extracted from experiments. So we connected to these data sets to actually build or construct a whole virion. And we've been working, as I mentioned, for some years. We have one tool developed called Lipid Wrapper, which allows us to develop these, or to construct all atom viral membranes around any type of geometries that you presented. So biologically realistic geometries and with compositions. So we created this whole viral membrane. And really, the single biggest obstacle to simulation at large scales is the membrane. And that's because differences in the curvature of the inner and outer leaflets create an enormous number of problems for molecular simulation. And it has to do with, at the end of the day, in order to run these simulations, you have to have numerical stability. And so the position of these atoms to get it correct down to angstroms turns out to be very difficult for these large complicated systems where you have many constituents, and it's not yet in equilibrium when you're building it from scratch. So we undergo, Abigail Dahmer and my group basically sort of has created a workflow where we start from sort of a membrane vesicle that we create, and then we sort of have to equilibrate it. And as you go through this process, again, because of the differences in curvature of the leaflets, you have different densities and so forth. And so you end up with holes and clashes that you have to fill in and resolve. And so it's this iterative process. And then you put in the spikes and the other parts of the virus, and you can actually, after a lot of work and tears you can actually get these big systems up and running. And so we were able to do that largely thanks to Abby, who's just a fantastic senior grad student. She's going to be graduating in December. But this is so cool to me. This is like, I think, so exciting because it really gives us now a whole new dimension for studying viral infection, and sort of and therapeutics, because we can now begin to really understand sort of what happens when these spike proteins are bunched up into their different, into crowded environments, and what kind of interactions does it have with, for example, glycosaminoglycans on the host cell and so forth. And so, so then just sort of a summary slide, I guess, of everything we did. So that everything I told you about was sort of packaged together into our Gordon Bell submission. And so this is showing the timeline last year from mid-February 2020, when those first images became available. And, these supercomputers, all the HPC providers were so generous in saying, do whatever you can. Scientists really collectively mounted a huge effort to try to understand this new threat. And we were among them. But going from early March when I first tweeted, and this is when I really just joined Twitter. I was like 2 days into Twitter and we tweeted this, and it was just like, couldn't believe it. So interesting. And then all the work that we did between March and October, when we submitted the submission and got this, the virus up and running. And I have to say, the actual science, so for the single spike system, we've gotten already a couple really beautiful scientific findings, a few, and they've been really helpful, for understanding all sorts of things. Lots of groups have reused this data. For the two parallel membrane system I told you about, and for the full virus, we continue to work on what are the actual scientific knowledge, what's the new science of the virus. That's going to be coming later this year, but I don't tell you about that today. Here I focus more on the technical challenges. And then just as a final slide, would like to leave time for questions and so forth. I just want to say, it's been such a special time, for everybody. It's been, I think, a difficult time worldwide. And it's been a special time for scientists. I think that maybe is one of the silver linings is that, how science has done a pretty good job of making a difference, of having an impact, certainly with the vaccines at least, and understanding, airborne transmission and so forth. But early on we realized, back in March 2020 and April 2020, as all of us as all the scientists, people in the field, started to assemble around this shared challenge, we realized we had to do things differently. Scientists tend to be very secretive about their work. We tend to hold our cards close to our chest. We don't necessarily disclose things before they're published. And, this is because of the incentive structure. Scientists are rewarded for being first. You get the science paper for being first. And then, everything else that follows is important, but it doesn't get the same kind of glamour treatment. And so, but we couldn't do this. Really, I think people really had to set their egos aside for this virus and just try to make progress and have impact, real impact, not just a paper in a high impact journal. So anyway, we realized that we needed to share things. And so I was so just happy to see all the people in our field, over 200 groups worldwide, agreed to a shared set of principles for working in the space, where we agreed to share models, methods, use preprint servers, et cetera. And that's really worked, and it's been amazing. And that Gordon Bell work that I just talked to you about, there were like 30 authors on that from all around the world contributing. It wasn't just me at all, or my group even. It was a lot of people, and that part has been really fantastic. And I hope that this sort of openness and sharing and so forth, that it continues on, to other areas also. I think that's going to be really important. One way that it might be able to do that. So this sort of movement in our field led to the development of a new way to share our data sets. So, in molecular simulation, we haven't been very good about sharing our data sets, and for a lot of reasons, not just the cultural ones, but also technically they're big data sets. Sometimes the weighted ensemble stuff I talked about, they're 200 terabytes of data. How do I give that to someone and actually have them use it? So this Molecular Science Software Institute, sponsored by the NSF, stood up together with BioExcel and other international partners, an awesome sort of networked data hub for everyone working in this space in COVID-19. And I think this is going to be a real legacy. One of the legacies of this disease is going to be this sort of this data set. And I think it's going to be really important for AI methods and for helping just the community in general do better about sort of like sharing and information and so forth. Okay, with that, I'd like to acknowledge it was a really big team and a lot of people coming together to do all this work, and here are all their smiling faces. Oh my goodness, there's so many people to thank. You know, the folks in my group, Lorenzo Castellino, Ziad, Amelia, Abigail, Brand, Mia, Fiona, Shirley, Terry, there's so many people. Also Jason McClellan and his team. I mean, what a terrific bunch of collaborators. Elisa Fada, the glycobiology community, also awesome. I mean, they are so inclusive. I think it's been such an exciting time. So they're just terrific. The AI folks, simulation folks, Arvind Ramanath and Shantanu Jha. And I don't have a picture here, I can't believe it, of Lillian Chong and her team and Carlos Simmerling and others who participated in the funding agencies. Also, here's a different view of all those folks with their faces on a virus. I'm happy to take questions, hopefully have some discussion. But thank you for your attention and for inviting me today to tell you about our work.
All right, thank you for a phenomenal seminar. That was incredibly interesting. And we already have questions pouring in the last few moments here. So I will open the floor for questions. I see there are 3 pending here in the Q&A. So if folks would like to continue typing them in there, or otherwise raise your hand and we'll acknowledge you to speak your question out loud. But I will start by relaying some of the questions that are typed here in the Q&A.
Okay.
So the first question here is there any transmembrane signaling of the spike protein, and does your model provide any insight into that?
Oh, that's just a great question. So to date, I would say I don't think we know the answer to that. You know, the membrane is a mysterious thing. There's a lot going on there that we can't get a really good view on. I will say, and I took this slide out, we are trying to see what might be happening there by using, so we have that huge, actually, maybe I'm tempted to show, since we have time, well, maybe I shouldn't. I'm tempted to show another slide from a different talk. We do have, we're using like a dynamical network analysis sort of method to see if there is communication or signaling happening in the transmembrane part. And so this is a way of looking at sort of small timescale correlation across, let me show you, I can pull it up, across these long time and length scales. So that is something that we're interested to look at, but we haven't, it's taking forever for this thing to load, but we haven't actually moved on yet. But let me just, I can show you a picture of some preliminary data there. And this is part of that work that, maybe you can see it now. So this allows us, using these 2 types of method, generalized correlation-based dynamical network, as well as this weighted ensemble, or this weighted implementation for suboptimal paths that I developed in my group, we can actually, so this is a side view of the spike protein going down the stalk to the transmembrane bit. Here's ACE2, you can see it there, and it's, you know, stalk and transmembrane bit. And we can start to look at if there are sort of correlated motions or signals that are, you know, sort of getting communicated through. So that's something that we're looking at, but we haven't published anywhere yet. And as far as I know, there's been nothing like really demonstrated.
All right, great question. Thank you for that part there. The next question is, are the spike proteins on the virion distributed in a specific way, i.e. certain distances apart from one another, or do the certain number of spikes vary on each of the virions?
Okay, so from the cryo-electron tomography, and there were 4 beautiful studies, as I mentioned, at the end of last summer that came out. These are, there's a lot of variation. They're heterogeneous. So these, you know, they obey averages, but there's sort of variation. So the diameter of the membrane of SARS-CoV-2 is about 90 nanometers in diameter, plus or minus 10. So it goes between 80 to 100 nanometers on average. Then you add the spike proteins. The spike proteins, there's on an average virus, there's usually about 25, and they're distributed basically randomly. But when you look at many thousands of averaged copies of viruses over these tomography studies, you can say like, okay, well, on average, they're each, 20 nanometers apart. But if you looked at, so when we build our model, we did also space them randomly, where they're not exactly like 20 angstroms apart. There's some that are bunched up and some that are more spaced out. That's a good question, though. Yeah, so there's a lot of variability, and that's generally how viruses are.
All right, and on a similar, I guess, more atomistic scale to that question about the spike proteins, does glycan formation on the viral proteins reach an equilibrium with the immune system, wherein further glycan protection would actually be disadvantageous for the virus rather than advantageous? The context being glycan evasion of influenza A and B and SARS-CoV-2, is it greater perhaps than viruses like measles that seem relatively easy to neutralize with a vaccine?
Well, I'll just say definitely one way that the viruses do work to evade, one tool that the viruses have, SARS-CoV-2 and influenza and many viruses, HIV, to evade the immune system is to, as they mutate, also introduce or delete glycans. And in fact, so I showed you that one example of the lab passaged virus from Rino Rapuli's group, where a glycan was introduced, and by introducing it, it kicked off like a whole section of neutralizing antibodies, like a whole group of them. But in the Delta variant, actually, it looks like maybe one of the glycans has been deleted. And that deletion also, because it's very close to where a neutralizing antibody is binding, that also, because it's sort of disrupted that interface, that is, they're modeled really, you're not modeled, but they're designed very exquisitely, right, for recognition. That manipulation actually probably is part of one of the reasons why also some of the neutralizing antibodies are less effective for that particular site. So yeah, we do see that. And there is this coevolution of the species and the virus and their glycosylation profiles. And that's a whole interesting area of study.
Next, we have a nice compliment from Marcus Gastrike. Goosebumps watching the presentation, and thanks for a great talk. So thank you for that, Marcus. And then the next question, could these models help an understanding of interactions between the RBD and ACE2 as presented in different tissues of the body?
Yes. Oh my gosh. I want to show, but I took this out. I always never know what to put in or take out. So one of the things that we are really excited about is trying to understand aerosol transmissibility. Do I have it in here? Oh my gosh, I took it out. I'm sorry. And so, yes, so we are working to build different models of different, basically lung surface cell types and different surface cell types. So, one of the really interesting features of SARS-CoV-2 is that it is in aerosols and it's infectious in aerosols. And Lindsay Marr, who's at your institution, I'm so excited to talk with her and her group later today. But we can use simulations to get views into what's happening in these aerosols, and also as they implant essentially or bind to these different host cell surfaces, particularly in the deep lung. So these small aerosols go into the deep lung, which has a particular, sort of set of expressed receptors. And yeah, we're working on that. That's sort of like, that's ongoing. So yeah, and it's cool because you can, these sort of models can not only be useful for like driving the development of experimentally testable hypotheses about like what is binding and where and what's controlling infection in various cell types, but also for the potentially to develop novel therapeutics, so that's like basic and applied knowledge.
All right, great question. Thank you for that one, Jim. 3 more here in the Q&A. There must be, this is the great question for those of us who do simulations. There must be conclusions from your model that you are less or more certain about. how do you deal with the uncertainty, I guess the confidence in the conclusions?
Yeah. Yes. Well, I didn't, and I didn't, because I was covering so much here, I don't go into the details of like how we, really sort of prove to ourselves what is statistically significant. I mean, we do that. So we sort of, we have many hypotheses at the beginning usually, and then, some survive, and some even, you're disappointed sometimes when it's like, oh, but it's not, it was such a nice story, but, I don't really think there's anything there. But I will say for us, the real test of our methods, in addition to all the theoretical ways that we can, compute statistical significance, but it's in whether or not these predictions turn out to be experimentally validated. And to me, that is like the ultimate test of the method. So that's where, we do the work to make sure that our hypotheses are statistically, meaningful. We do that up front on our own way, because, well, that's important because I don't want to go to my experimental colleagues and tell them something that, was really just completely fabricated because it wasn't significant. Because that, sort of reduces the trust the next time when I actually have one, right? So we try to do our homework up front, but at the end of the day, it's really, to me, the proof is in the pudding when you put it to the test in the real world. So, yeah.
And then regarding structures for the information that we use to start the simulations, in your experience, what are the major differences between structures obtained from cryo-EM versus traditional X-ray diffraction?
Well, a lot of times at the end of the day, right, we're ingesting just the position of the atoms, so it's the same for us. But of course, with cryo-EM, it's allowed us to get much bigger systems that were otherwise sort of, incalcitrant to crystallography. So that's been awesome, like the system type and the size, and also like groupings, like macromolecular assemblies. I'm all about, as you can see, like assemblies and, making things sort of more complicated. I think complexity is amazing. So that's certainly one big difference. But it comes at a price, right? So we also care very much about chemical matter. And so, you need to know exactly where the atoms are in a small molecule. A lot of times you can't get exactly down to that resolution with cryo-EM sufficiently, and you need to move to something like crystallography where you can really see more precisely, the positions. So, I think in the best cases, as I try to impart, we're sort of synthesizing multiple different types of data sets, structural and otherwise, to converge to developing these sort of most accurate, maximally accurate, or informative models of these systems.
All right, and our last question that we'll have here, we've gone a little bit past one, but that's great. Discussion's been wonderful. The last question is, does the spike protein bind with the endogenous ligand binding site of ACE2? And if it is, why is it doing that? Is that because ACE2 is primed for grabbing something from the air, or just is the virus exploiting some other feature of ACE2?
I think it's mostly exploiting another feature of ACE2. ACE2 is an angiotensin converting enzyme. It's like involved in clipping like renin. It's like and like blood pressure regulation. So it's doing something else. It's actually an enzyme inside there, which I didn't talk about at all, which is kind of horrifying because I love enzymes. But I kind of ignored that feature of that system because we're talking about just binding on this sort of other interface. I think that's so I think it's predominantly exploiting this other part of a biological molecule than what it's like ordinarily doing. That's a great question.
All right, that's our Q&A. Thank you everybody for a very engaging discussion and for coming today. And please join me again in thanking Dr. Amaro for a very stimulating seminar. And everyone have a wonderful day and a nice weekend.
Thanks for having me.